the synapse now. This is, yay, review, because we've done this before. We're just going to show it in a little more detail and in the context of from nerve to nerve. A synapse is going to have two different cells. One of them has to be a neuron. We will have a neuron that's going to send an action potential down to its axon terminals, and it can go to another neuron, like in this picture, or it could go to a muscle, like in the example we've done at the neuromuscular junction. In essence, you have, after an action potential, we're going to talk about what's going on at the synapse. And we already know, big picture, chemicals are going to get released and there's going to be a receptor on the other side. So we'll just go into this in a little more detail. So the players of this are the presynaptic cell, that's sort of the neuron bringing the action potential down. Then we have the postsynaptic cell, which is receiving. It could be another neuron, or it could be a gland or a muscle. It could be anything that's going to be getting this message to do something. Between the two cells is a synaptic cleft, the gap. So what's this picture? I really like this picture because you can see how the axon terminal of one neuron is not physically touching the second neuron, but instead we'll sprinkle it with different types of chemicals. We'll go through the list in a little bit, and there'll be a receptor there. So we're going to go into detail here. Another picture here. So the expanded ends of the axons are known as axon terminals, synaptic knobs, synaptic bouton. There is lots of different ways to describe it. They are going to contain little pouches, which are vesicles that contain neurotransmitters. The neurotransmitters are chemical messengers. Once they leave, they can activate a sodium channel. If they bind to a sodium channel, what's the effect going to be on the cell? Depolarize or repolarize? If a chemical messenger, one of these chemicals, gets released across the cleft, and the other side it binds to a receptor, and if the receptor causes it to open a sodium chemically gated channel, what does that affect? It will depolarize. It's going to make it more positive. Some chemicals can go and bind to potassium channels, and they'll cause that to open, but then potassium will leave. And that would cause the cell to be what? Hyperpolarize and take it further away from an action potential. So here's my little diagram where we have a super zoomed in view. This is the axon terminal with vesicles with little red neurotransmitters from one of the neurons going to a postsynaptic cell. If it's a muscle, this would be sarcolemma. If it's another neuron, it could be a dendrite. These here would be channels. So the neurotransmitters are going to be in red. The receptors for those neurotransmitters are on the other side. The red in the membrane there sort of denotes the action potential from this presynaptic neuron arriving. That causes these vesicles to come to the edge, fuse to the edge, and ultimately spill out their neurotransmitters. These neurotransmitters will then bind to the receptors on the other side of this cleft, the synaptic cleft being the gap. Or the neurotransmitters bind to these receptors. It will, if it's excitatory, only if it's excitatory, which means if it's going to be a sodium one, it will depolarize or activate that postsynaptic cell. This is what happens to a muscle cell. Acetylcholine bound to receptors on the sarcolemma, causing actually sodium to open, and it became more positive. And then an action potential went around the sarcolemma and down the T-tubules. So in this case, we have sodium out here, and you can see where sodium is now travels across the membrane, and now we have more sodium inside the cell, so it's depolarizing, becoming more positive. And it's usually a one-shot deal. Once we release the neurotransmitter, it's out there, and it doesn't just stay hooked up. We don't want it to keep endlessly stimulating this nerve. So what we have are these little enzymes that's going to break apart the chemicals that are bound to the receptor. These enzymes are going to pluck off the chemical. If we're talking about a muscle, it'll be acetylcholine, and the enzyme is called acetylcholine esterase. So I'm not having you know that. I just want you to know ACE, remember ASE is the ending that implies it's an enzyme. So in this case, we have these yellow Pac-Mans that are actually plucking off the neurotransmitter, and they will actually break the neurotransmitter apart, so it becomes no longer active. So it's a deactivator of these neurotransmitters. So now we have these little scattered debris field of broken down neurotransmitters. So therefore, our postsynaptic cell isn't getting pummeled and re-stimulated if we didn't really want it to. But we like to recycle. So we bring in, reform these little particles and put them back together, and they end up reforming back into the vesicle so they could be used again. So that's really what's happening at the level of that cleft. Not only do we have an action potential that sends out the neurotransmitters, it's received on the other side by receptors. And in this case, they open up sodium channels so it could be more positive, but it could do it to other channels as well. On the receptor, we have an enzyme that plucks it off and chops it apart so that we don't have, so that we stop activating our cell. We bring back the individual parts, reassemble them, and put them back in a vesicle so it's ready to go again. So this is our recycling process. This is in your notes. It's just a list of them step by step of all the stuff we just did. Neurotransmitters. Now at least you know where the neurotransmitters are found. They're in vesicles, which are found in what part of a neuron? Axon terminals, or many of the other names for it. Excellent. When a neurotransmitter is released from an axon terminal, what space is it crossing? Synaptic cleft. And where is those neurotransmitters going? What are they going to bind to?
Channels, the sodium-gated channels.
They're going to bind to receptors. And technically, those receptors are now chemically gated channels. The fact that a neurotransmitter is a chemical. So you're binding to receptors of these chemically gated channels. They could be sodium. They could be potassium, they could be chloride, they could be other ones. We were just learning about sodium and potassium. So I really wanted to make that point that sometimes they could make it depolarized, sometimes they can take it away from depolarization and make it harder. There are two main classes of neurotransmitters. Some are excitatory, those will stimulate sodium. Some are inhibitory, those are going to stimulate potassium. The effect of a neurotransmitter depends on where its receptor is. Is it on the sodium channel or is it on the potassium channel? The point I want you to think about is a single neurotransmitter in one place of the body might actually activate a cell. But in a different place, it could inhibit. It really is about what's going on with the receptor in the different locations. So what we're talking about here are chemically gated channels. On this diagram, you could see the channel is closed. The red dots are just the ion that you're keeping out. We have a dark green neurotransmitter that bound to the channel, opening it so that the ions can come in. So again, it's only triggered if a chemical was binding to it. So the types that we're going to go through are small molecule and neuropeptide. I put in the categorization of amines versus amino acids, but I really don't need you to know these subgroups. I've sort of just listed there for organization. Neuropeptides, we will talk about at the end because they're a little bit different than the whole recycling thing that I just talked about. So all these that we're going to do first, acetylcholine, norepinephrine, dopamine, serotonin, GABA, and glutamate are all going to function exactly like I just told you. It's going to get released, bind to a receptor. You'll have an enzyme that breaks it apart. You have the broken up pieces, go back up. We've put them back together and reform vesicles. They do that. The endorphins and substance P are neuropeptides. They don't. So we'll just put those at the end. Acetylcholine. So now we're just going through this list. Acetylcholine is something that you're already familiar with. So we know that it was at the neuromuscular junction. So we know that a motor neuron going out to your body is going to send out acetylcholine onto muscles, and that's what's going to stimulate muscles. Also, acetylcholine is also utilized in the brain but it's in their memory centers. So if somebody is having problems with their acetylcholine, not enough release, or maybe something's damaging the receptors, then you're going to be on the path to Alzheimer's disease. So Alzheimer's disease is a hallmark disease for acetylcholine problems, but within the brain. Has nothing to do with them not being able to move. So that's why I say just because it's a chemical, it's really more about where they are in the body. So within a specific part of the brain, the hippocampus, if you have problems with your acetylcholine receptors, you're going to have problems with long-term memory. Examples, but the main one I want to focus on that I probably would ask a test question about would be the Alzheimer's disease being the pathology associated with acetylcholine. You can also have, though, what's known as myasthenia gravis, and that's an autoimmune disease, and it's actually attacking acetylcholine receptors on your skeletal muscles. And so people there with that disease have a difficult time with their motor function. Drugs like nicotine or curare even, curare is like an Amazon drug, comes from like the crazy poisonous, frogs in the Amazon that you put on the tip of the spears if you're an Amazon native and you're going to shoot some sort of wild boar, and then your arrow might not have killed it, but the poison on the arrow sure does, because what it does is it actually blocks acetylcholine receptors, and the boar drops over dead because it's paralyzed and it can't breathe. So that's actually more effective than the arrow itself was in that case. The bottom line for acetylcholine that it sort of as a summary is if you know acetylcholine, know that it's used to stimulate muscle. So it's at the neuromuscular junction in the brain. It's part of the long-term memory, and a pathology associated with acetylcholine would be Alzheimer's disease. That would be the highlights of this one. Norepinephrine. So norepinephrine is a neurotransmitter in the brain. In the brain, neurotransmitter norepinephrine initially is a mood booster. It makes you feel good and it makes you feel happy. Too much is not good. Amphetamines are actually going to increase the release. You're gonna have extra norepinephrine. People that take some of these drugs feel good, and that's where it can be addictive because maybe they're feeling really bad and now they feel really good. Cocaine will block the removal. So remember, the neurotransmitter goes in and we have an enzyme that breaks it apart to make it stop activating, to deactivate it. Well, cocaine says, you know what, I'm gonna get in the way of these enzymes so that whatever norepinephrine you already released, it's just gonna be more effective and keep stimulating. So it's the same effect, you're still feeling good and happy. However, too much norepinephrine can actually overall increase aggressive behavior. So we have that. So norepinephrine at the base level is one of the three that we're gonna be talking about that helps alleviate depression, makes people feel better. Norepinephrine, though, too much is just too much. So norepinephrine in the brain is your feel-good neurotransmitter, but too much increases aggressive behavior. Amphetamines are gonna make you release more. Cocaine is gonna block the removal and therefore hangs out longer. So both of them will be the same in terms of its effectiveness and on the person of increasing their responsiveness to norepinephrine. Dopamine, it's our second of our happy, feel-good neurotransmitters. Dopamine has two jobs. First job, makes us feel good, makes us happy. Dopamine, and not just feel good and happy, it's satisfied. That's probably better, so you feel content and satisfied. So again, that's another element that's in depression. But I wanted to point out this satisfied, content feeling in people that don't have enough dopamine release. If you do, sometimes, some people, if they do more like high risk sports, they want to fly out of airplanes or, a lot more things that require a little bit more stress that some people will, you're nuts for doing that someone really gets this adrenaline junkie kind of people. They tend to be more dopamine deficient. And so this high risk behavior makes them feel good because it increased their dopamine release. And then they get this so that makes them feel content. Might scare the beans out of somebody else. But for these folks, it makes them feel a little bit more content. It's also why kids with ADHD, it's a dopamine deficiency problem. So kids with ADHD are fidgety and they're moving around and they're just jumping up and down, and their movement is them trying to stimulate this release. So in fact, giving them ADHD medications is often some sort of stimulant to other people. It stimulates the ADHD child sufficiently that they get the dopamine that comes out, and then they can have a contented feeling. So it's like they're malcontented and they're running around until they get their dopamine release and they're feeling satisfied, and then they can focus. So there's a level. So I want you to be connected that dopamine is a neurotransmitter that helps us feel good. So it's one of our depression ones, but more on a satisfied note. Its pathology would be ADHD. So if you don't have enough release. In a different part of the brain, which is why it's in a different color on the slide, in a different part of the brain, known as substantia nigra, it's involved in muscle coordination. So the part of the brain that is involved in muscle coordination, if it doesn't have enough dopamine, you're not going to be able to coordinate your muscles very well. And the pathology associated, because it's a very specific part of the brain, is Parkinson's disease. And you'll notice if you see someone with Parkinson's disease, their motor gait is they will walk kind of like a robot. They're a little R2-D2-ish. And they're doing that to make them more stable, and each step is more purposeful. And if you see them, and you don't know if they're just maybe afraid if they fall, they don't want to break a bone, you don't know if they're just being careful, but usually the trick is, not the trick, but really the definitive sign of, oh, it's Parkinson's, is if you see them turn, and their turn, this is the R2-D2 part, they turn like this. They're not able to just go and swing their leg around and just go that way and change directions. Not that we're like all Fred Astaire walking around or anything, but they're not able to do that. And that's this motor coordination. So they get treated with additional L-DOPA. We're just infusing these guys with some L-DOPA, and it helps them out a little bit. It increases their dopamine. So I want you to associate dopamine with a feel-good, satisfied, pathology is ADHD, or Parkinson's disease if it's the specific part of the brain that you're having dopamine neurons that are getting damaged, and therefore you'd have muscle coordination problems with Parkinson's disease. We can see also over here amphetamines and cocaine. It's the same pattern. Amphetamines are going to increase the release. So again, that's why people get addicted to drugs. It's a feel-good neurotransmitter, now they're getting more. Or cocaine is blocking the reuptake, so it's hanging out longer and is more effective. So we see that pattern again. Serotonin, our third feel-good neurotransmitter. So it makes us feel good. Serotonin's also the main one that gets treated when someone has depression. So if you are taking Prozac, or someone's getting prescribed Prozac, it's known as SSRI. Does anyone know what that SSRI stands for? Yep. Selective, so it means it's a really specific one. Serotonin, so selective serotonin reuptake inhibitor. So what does that tell us that it does? It tells us exactly what it does. Why don't you explain it to me what it does? It raises the amount of serotonin that's out in the cleft, and it didn't affect the release. You blocked the reuptake. So you prevented it from coming back in. It's hanging out into the cleft longer so it can continue to stimulate that other neuron, allowing serotonin to be more effective. So I want you to realize serotonin, Prozac targets serotonin, makes you feel good. The pathology obviously associated with that would be depression. It also has insomnia, appetite loss, all those things really go with it as well. Glutamate, learning and memory. So glutamate, and when we say learning, this is more, let's just say, laying down memory. So this would be glutamate helps you with your long-term memory. That's what learning is. You're sort of putting something so you can recall it much later. So not only is glutamate helping you as you get new information in, you want to make sure there's enough glutamate around so that you can sort of file it away in the place that you can retrieve it. And now that's what learning is about. Alcohol can impair glutamate, can inhibit glutamate. Glutamate is involved in memory and learning. Learning is really long-term memory, essentially. However, if you're going to drink some beers while you're studying, don't do that, because you're going to impair your glutamate. You're just going to waste your time. If you know, again, with this sort of anecdotal extreme case, if you're out there with your buddies drinking and you know, you're like, oh, here's this new person. I'm going to meet Susie, and I'm going to meet, you know, Kim, and I'm going to meet, you know, you're naming these people. And then like, what was her name again? You know, you don't remember anything because you're drinking. Like, or if you drink too much, you're like, what did I do last night? You know, there's no memory. So that is how alcohol can impair glutamate at that level, if you want to think of it that way. GABA. Now this is one we'll all just agree to go ahead and stick with the abbreviation because it's a GABA aminobutyric acid, but everyone just calls it GABA. It is our main inhibitory neurotransmitter, which means, what do we know about that? Which channel is it going to open up? Potassium. So it's inhibitory, so it's actually going to hyperpolarize the neuron. It is involved in a couple of things. So we'll first do the inhibitory side. Its pathology is epilepsy. So if you imagine the neurons in this particular areas of the brain that we may normally, in a normal sense, have some GABA around, that we kind of keep at a lower, you know, keep our resting membrane potential down. And if we want to then move or create a stimulus, obviously we'll depolarize it. If you don't have enough GABA, that person's resting membrane potential might not be -70. It could be -60. So barely any graded stimulus could cause it to reach threshold. So what I'm saying is, if there's a person with not enough GABA, their resting membrane potential is going to be higher because GABA is an inhibitor. It keeps it nice and low. We good with that? And so if you don't have enough, if this is a pathological situation, you're closer to threshold, it means any little thing could likely cause that neuron to fire as well as additional neurons, which would cause a seizure. And that's where epileptic patients respond or have these triggers that would not normally trigger other people. So their medications would be more GABA to lower their resting membrane potential so their neurons don't fire so easily. If you have a person that's not epileptic and you're just taking Valium, you're just a little slow to respond, right? Because you're depressing the nervous system by making it harder to get stimulated. The other part of GABA is movement. With movement, this is the other example, and I don't mean to be harping on alcohol, I'm just using this as sort of an example. You know, people that if they're drinking too much alcohol, not only can you not remember Susie's name, but you're going to be walking kind of funny, right? So people that are drunk, they're going to be not as steady, you know, and their feedback mechanisms are a little more like, whoa, I went over too far. I need to overcorrect now. You know, like you can see the responsiveness is going to be slow, but they're also having difficult time with motor function, you know, like trying to put the key in the door or like dial their phone or, you know, like that's going to be difficult. So you can see that alcohol can also impair with GABA if GABA's movement functions. So it depends on what part of the brain we're talking about. You know, it's going to be a different part of the brain than would be the epilepsy that would be triggered. But again, it's associated with movement. Alcohol can impair it, and you're going to lose motor skills. So looking at the ones we just talked about, acetylcholine, norepinephrine, dopamine, serotonin, GABA, glutamate. We know that norepinephrine, dopamine, serotonin, those are the three associated with depression. We know that acetylcholine is associated with memory, long-term memory. So it's in a specific part of the brain. So loss of acetylcholine function in the brain leads to a pathology of Alzheimer's disease. Systemically, acetylcholine is a neurotransmitter used to activate muscles. So if you gave an acetylcholine receptor inhibitor systemically, you can cause paralysis. So glutamate, GABA, who can remember what GABA does? It's our inhibitory neurotransmitter. So it keeps our thresholds low, and it takes us further away from depolarization. If you have a problem or a pathology associated with GABA, it would be epilepsy. You could also, the motor function, GABA has some motor function as well as dopamine. We'll talk about dopamine next. But GABA's motor function, if you have alcohol, will impair your responsiveness and affect motor function. Dopamine is one of your feel-good neurotransmitters, but it's mostly associated with satisfaction. So ADHD is the pathology associated with dopamine. The last bit that we're going to do are neuropeptides, and I told you they act different. The normal acting neurotransmitter, we know that gets released, hits a receptor, gets broken down, we bring it back and recycle it. Neuropeptides are not that way. Neuropeptides are, here's a neuron, and we have the axon, we have our little axon terminals, you know. What a neuropeptide does is it makes, the nucleus is up here, so it makes its neurotransmitter vesicles. It packages it there. And each day, you know, it's moving along and it moves everything down to finally bring it down here. Okay, that's all, a lot of work. It takes a long time to make this happen. And then when it's finally going to be released, say to another neuron, here we go, this way, it's going to be released and we let it out. We just let it go. We don't recycle it. We don't do anything. It just goes. And then we have to make a new batch. And it takes several days to get it down and then to come over here. So it's really not one that we use on a regular basis, that we're letting out, bringing back, letting out. It's really one that when it's let out, it is longer acting, long sustaining effects. But because when we release it, it sends a signal to make more and send more down. So upon repeated use of this, the neuron's like, I better increase my production. So it starts to make more. And so then in a way, you want to release more. Does that make sense? So we'll go to the first one, endorphins. What do we know about endorphins? Makes you feel good. How do you get endorphins? Exercise. So you know those exercise nuts, those fanatics that just exercise all the time? They are getting their endorphins and it's making them feel good. Now, if you're starting an exercise program, saying the endorphins take a while to kick in. So you might go, it's like, if I decide, okay, I'm going to start running again. Okay, I better start running again. So I get my running shoes on, I go out, and I'm like, that was not fun. And then, you know, so just like you have for sheer perseverance. I always tell people, if you're going to start a program, give yourself 2 weeks. 3 weeks really, but you know, like 2 weeks, you're already invested that you can probably get the 3. After 3 weeks, you're like, you know what? It's not so painful. Like you got to go on sheer willpower the first 2 to 3 weeks. But after that, you're like feeling pretty good. And then it's beyond that, the people that run and get greater intensity and longer durations are getting more of these endorphin releases, because the endorphin is like your natural morphine, and it actually pain reliever, it actually makes you feel good, light mood. So then that person that's used to these long, hard runs and these endorphin, really a daily endorphin dose, is like maybe on a road trip and can't go anywhere and is stuck. They're going to get really agitated and they're going to have a problem because they're now addicted to this release, and their production of endorphins are going to be built up. So that's where you get some of these people get a little neurotic. I'm teasing if somebody's out there. My father is like that. He's a complete exercise nut. So the point is, I want endorphins to let you realize, as a neuropeptide, it's made here, released down there, we don't recycle it, and then we can increase our production of them the more we ask to use it. So that'd be like increasing your exercise demand over many months. So endorphins are feel good. Substance P, I like to name it as substance pain. Substance P is pain. if you have damage, something causes damage, substance P is released, and it's slow to build up because it's almost like the neurotransmitter trickles out. So it has to do with pain, duration, and intensity. Probably the best example is like a jalapeno. You know, you put like a hot, at first you're like, it's not so bad, you know, and then it starts to like increase in intensity. That's because it's releasing substance P and it's now increasing its intensity. So it's the opposite of the endorphins. They both are made and work the same way. They're peptides. So I put the two together for you to learn. Endorphins is sort of the opposite of your substance P. Substance P is pain, slow to build up, but hangs around for a long time. Again, with the jalapeno, you might have like took a bite, your mouth is on fire, and it's gonna be on fire for a while, even though it's long gone and you spit it out or whatever, where you have the endorphins to combat it, but not always. Sometimes it's more of just that, not because of a release of substance P. Sometimes it is. So substance P is released in with and is associated with pain, and long duration increases in intensity. Endorphins are released to combat substance P. So you have a balance of the two. So here's endorphins. Its first job is to inhibit substance P. Intense exercise will increase the release of endorphins. Also, opioid drugs. That's why people get addicted to them, because they fit our receptors perfectly. And so it is our unnatural way of getting our endorphins is through opioid drugs, morphine, heroin, and those. In the brain, it's going to increase GABA, and an inhibitory effect, but also can increase pleasure, the dopamine side of things. So you can see where some of these drugs can become very addictive because your receptors get used to having them, and then it's also giving you these other feelings as well. So that's all I want you to know. Here's the list. Essentially, so those are the list here. I want you to know, substance P. What is its job? Pain. You can have mood changes and vomiting if you're in severe pain, not GI issues, but more severe. Someone who's in such pain they had vomiting. So that could be extreme case of capsaicin would be just more like the chili pepper kind of hot thing. Endorphins are the opposite, morphine and exercise, and then we have some of these others as far as the modifiers or what's you need to kind of trigger. So you should know the effect of alcohol and things. You should know the effect of amphetamines and cocaine. Essentially, where amphetamines will increase the release of some of our neurotransmitters, where cocaine inhibits the reuptake. And it's not so much that I care about amphetamines or cocaine, it's still that I want you to get the process. Sometimes they make more and send more out. And I know that it comes back, and if you block the bringing back, you're increasing its effectiveness. So it's less about being focused on the drugs and more about how does a synapse actually work. So the drugs are just easy to utilize as modifiers of that. So that's why it's not that I'm trying to be drug focused here. But that's it. Yes.